recording.
Actually, yeah. It's just right behind you. No, yeah.
There now?
Yeah. Okay, so
I guess I should just.
raise your hands if you know someone who is suffering from or who has suffered from mental illness. Okay. Raise your hands if you think basic research in mice has anything to do with understanding mental illness in humans. Okay. Well, as I said, I'm Darshini. I'm a fourth year PhD student in Mark Ansel's lab, and I plan on spending the next 20 minutes or so telling you about how I utilize mice in my research to understand the driving forces behind aggression as a pathological or a disease-related behavior. Can you see, Morgan?
Oh, yeah. Yeah.
You're welcome to stay here. Okay. So we see it in the movies, we read about it in books, and unfortunately many of us experience this in person, but we all know that mental illness is associated with a great deal of suffering. Unfortunately, not very much is known about the underlying causes and so subsequent treatments of these as well, at least in comparison to diseases of the body. So consider ourselves, we're in 2016 today, and if someone goes to the doctor with a concern of a cancer diagnosis, they're sent off to get blood work, biopsies, bone scans, the works, right? But if someone goes with the concern of a depression diagnosis, they get a piece of paper, a questionnaire. And there's like something fundamentally wrong with that. And the problem lies in the fact that we have an overtly simplified view of the biological basis of mental illness. Now, so far, it's often been viewed as being, these illnesses have been viewed as being a chemical imbalance in the brain, as if the brain were a hot bowl of soup filled with chemicals or neurotransmitters such as dopamine, norepinephrine, and serotonin. Now, neurotransmitters are those chemicals that are responsible for communicating information from one part of the brain to another. Now, we can't be entirely to blame for thinking of it in this way, because a lot of the drugs that are prescribed today to treat mental illnesses, such as Prozac, which is used for depression, act by changing the brain chemistry throughout the brain, as if the brain were in fact a bowl of soup. Now, unfortunately, these drugs don't work that well, and many people can't often take them because they end up producing a bunch of different side effects. And the fact that they do is not entirely surprising because they've been used to treat very complex psychiatric disorders. And using them to treat these disorders is almost like trying to change your engine oil by opening a can and pouring oil all over the engine block. Now, part of the oil is going to go into the place you want it to go into, but a lot of it is going to go all over in other places as well and cause a lot of harm. Now, the field is beginning to move forward and think of the causes of mental illness in a different way as being brought on by disturbances in the functioning of neural circuits. Now, circuits are those connections that connect the different parts of the brain, okay? And these circuits are responsible for controlling moods, emotions, and cognition. Now, when we think of cognition, what comes to mind very frequently is the very popular computer analogy. So we have computers made up of a bunch of different wires that come together to perform a function, very much like the way our brains function. The truth is, we can also use this computer analogy to understand how our emotions work. We're just not wired to think of emotions in that way. Now, not very much is known about this circuit-based hypothesis of mental illness. It's fairly new, and it's also largely because of the dominance of the chemical imbalance hypothesis. It's been around the block for very long, and I'm not trying to say that chemicals are not important. They're very important. They are the messengers of our mind, and they're responsible for communicating information throughout the brain. It's just that they don't bathe the entire brain like soup, okay? What they really do is each of these chemicals act at very specific locations in the brain in particular regions to produce their effect or to alter the flow of information. And to truly understand the biological basis of a mental illness, what we need to know is where each of these, or pinpoint where each of these chemicals are acting, the regions they're acting in to affect a change, to produce a change in behavior, so that we don't pour oil all over our mental engines and suffer the consequences of that. So you may ask, how do researchers tackle this complicated question of understanding the brain's intense circuitry? And the way we do that is by using what we biologists refer to as model organisms, such as the fruit fly, the worm, and the mouse. And these mice actually, these models are fairly similar to ours in many ways, such that we can draw very useful conclusions by conducting experiments in these. They allow us to use powerful techniques that enable us to molecularly identify the different groups of or populations of cells in our brain. And once we've done that, we can take a given population and play with the activity of just that group of cells, either by causing them to fire more or by blocking them from firing completely and seeing how this impacts behavior. Okay? So if we block a group of cells, such as the dopamine cells right here, and we block a behavior, we can conclude that these cells are necessary to produce this behavior, right? But if we cause these cells to fire more and we produce the behavior, we can conclude that these cells are sufficient to induce that behavior. So how does this pan on to what I do? So what I do is I look at the role of this particular population, the dopamine cells in our brain, in controlling adolescent behavior, which is in the mouse about a month to a month and a half, and this roughly maps onto the teenage years in the human, and adult mouse behavior. Now, mice, like humans, have dopamine in their brain, and it actually acts in a very similar fashion as it does in our brains. And it controls multiple different behaviors like attention, arousal, reward, and disorders of this system can often be associated with drug abuse, Parkinson's disorder, ADHD. But the behavior I'm looking at specifically is the role that dopamine plays in controlling aggression. Okay? Now, you guys may ask me at this point, and I think a lot of researchers who work with these models ask this, a lot of people ask researchers who work with these models the question that, how can an organism like a mouse teach you anything about an emotional state? How do we know if the mouse is aggressive or not? With humans, it's really easy for us to do this, right? We infer this based on facial expressions. We look at a person's eyebrows, the eyes, the mouth, and together all of this helps us to gauge what a person is experiencing. Unfortunately, this is pretty hard to do with mice, right? The truth of the matter is, no matter what emotional state they're in, they always look this way, right? So it's pretty hard for us to actually work around this, but we do start at some point. And the way we circumvent this problem is by taking an emotional state, defining the general properties of that state. Defining the general properties of that state, and then identifying behaviors in our model, the mouse in this case, that reflect those properties. Okay, so consider as an example aggression, which is my behavior. It's defined as an overt, violent behavior that's enacted with an intent to harm and or cause physical injury to another individual. So keeping these properties in mind, there are a few models in the field used to test, or paradigms used to test aggression, but the one we use is called the isolation-induced aggression test. Here, the two mice are kept in one cage for a period of 10 days, and they're separated by a perforated divider. So they're technically existing in isolation right here. The perforations in the divider allow them to see each other, hear each other, and smell each other, but they can't touch each other at all. The idea is over the 10-day period, they get revved up and they begin to develop a sense of territoriality of their cage space. And after that, I take the divider out and observe them freely interacting. Now, keeping in mind what I just said about modeling aggressive behaviors, do you guys have any ideas of what behaviors we may look at to model aggression in the mouse?
Do they fight with each other?
Exactly, yes. And anything more specific that might happen in the fight if you were a mouse, can you imagine?
They fight one another.
Exactly. Yeah, exactly. And so, keeping like exactly what you said. So the things we actually look for is the amount of time a mouse spends biting, as you correctly pointed out, the other one, or tail rapping. Now, this is like a rapid movement of the tail that goes this way that indicates like an intent to attack, that it's about to start fighting, and attack mounting when it basically jumps onto the neck of the other mouse.
Usually cats, when they wag their tail like that, they're going to play something.
Yeah,
too.
exactly. Yes. So it's like, I guess it's something that we see in like different animals where, you know, it's like indicates like an intention to go for it. And so it's the summation of all of these different behaviors that we consider our output like of aggression. So this is our aggression parameter. And so coming back to what I do, I want to look at how dopamine controls all of those different behaviors. And the reason I decided to work on this is because in our lab, we found that if we increased dopamine firing in the adolescent mouse, again, this spans on to the teenagers, maps out to the teenagers and the human. If we increase dopamine firing during adolescence, this made the mouse significantly more aggressive in adulthood. Okay? So to walk you through the timeline, the mouse is born, and then during adolescence, every day we give the mouse an injection with a drug called GBR, that causes an increase of dopamine firing throughout adolescence. Now, GBR, I should say, acts very much like cocaine does in our brains. After that, the mouse is just left alone, and in adulthood, we assess levels of aggression using the paradigm I just described. And what we found was that the animals that were given GBR to have increased dopamine firing throughout adolescence were significantly more aggressive. So here we have what we call the y-axis. It's like the axis that basically shows our measure or level of aggression. And so the GBR injected mice were significantly more aggressive than the animals that had nothing done to them during adolescence. Yeah.
And are adolescent, do you measure aggression in adolescence as well? Or is there a reason you wait until adulthood?
Yeah, because actually adolescence is a time where I think we like it. The behavior isn't that established just as yet, and a lot of the fighting can be like confused because there's also a lot of playful behavior that happens between the animals, and so there's like a very fine line between like play fighting and like unprovoked aggression. And so during adulthood, we find that this mouse that was given GBR during adolescence is significantly more aggressive. So the reason this was of like interest to us is because it showed us an association between an overall increase of dopamine and an increase in aggression. But as I said in the first part of my talk, we can't just increase dopamine throughout the brain and say that this is impacting this behavior. To truly understand what's causing this behavior or the role this chemical is playing, we need to pinpoint that specific location in the brain where dopamine increase is leading to this behavior. And to do that, we use a technique called optogenetics. This is a slightly complicated technique, so please feel free to stop me if you have any questions. But think of it almost as a on and off light switch that controls the activity of a group of cells, okay? And for this, the opto refers to light and the genetics refers to the genes of our mouse, our mutant mouse. And the reason it's a mutant is because it's been genetically modified, okay, to include a certain property. So specific cells, so in my case, the dopamine cells, gain a channel. This channel is basically a light sensitive channel, and only the dopamine cells are genetically modified to gain this channel. All the other cells are not. Following this, we use visible light that we shine into the mouse's head, into the brain, specifically in one region. So I implanted just at one region surgically, connect the mouse to a source of light, and then shine light just on that region in the brain. Now the cells are not all going to respond to this light, only the cells that have been genetically modified to gain this channel. What happens when we shine light is the channel opens up and a bunch of positively charged ions jump into just these cells. Because there's no channel here, there's no way for them to get in, Now these cells get revved up and charged up, and they fire to release dopamine, and then we test what happens to the behavior in real time. So in this way, we're able to gain specificity just targeting one group of cells, in this case dopamine, by using this light-sensitive channel, leaving the others unaffected. Okay? Does anyone have any questions? Okay, cool. So how does this work in aggression? So as I mentioned, my hypothesis is increasing dopamine increases aggression. So to test that hypothesis, of course, I should start with the region that produces the most dopamine cells. And this is a region we call the VTA, the ventral tegmental area. And so my first course of action was to directly implant, surgically implant this fiber optic ferrule, which is, I'll show you afterwards, it's how we deliver light into the brain, and stimulate it such that the light falls only in this region, and only the dopamine cells in this region will begin to fire out, right? And what we found in terms of the behavior when we measured aggression, when we did that, is that the mutant mice that had that channel, okay, to have a direct increase in dopamine, were significantly more aggressive than the control. So these are animals that are untouched, they don't have that channel, so they're not going to respond to light, and they have a very low level of aggression in comparison. So this experiment allows us to draw a more direct cause and effect relationship between this group of cells and this chemical and this particular behavior. A manipulation that's almost impossible for us to do in humans, right? To gain such specificity. Now, this is what actually it looks like in the mouse when we implant it and into a specific region and shine light. And then what we do is we get the brain to see and cut the brain. And this is an image of a 40 micrometer thin section that is achieved from the brain. The bright green here is the region VTA, and the cells are the ones that get activated when we shine light. The little tear over here is achieved because of like the surgical implantation of the fiber optic ferrule. Now we're stimulating, or we're causing dopamine to fire out from the VTA, but there are multiple places in the brain this dopamine could go to. And in order to like better understand which circuit is responsible for this, we have to look at all the target regions that the ventral tegmental area sends dopamine cells to. So if you can see that I have the same diagram that over there, and we have regions A, B, and C, which the dopamine projects to. Now, when we stimulate directly at the VTA, all the dopamine cells right there have sodium rushing in, and they release dopamine at all their different target points, right? But we don't know which one of this is producing aggression. The way we answer that now is by taking 3 separate groups of mice and doing 3 separate sets of surgeries where I implant my fiber optic ferrule into the separate region specifically and stimulate the cells only in that area. So tackling this one at a time, I started with region A. As you can see, I've changed the location where I've implanted it such that only that area gets the light. Dopamine is released only on region A, and then I measure if this increases aggression, and I found that it did not. Then I moved to region B and stimulated deeper in there. In region B, tested for aggression, did not find any changes, right? Finally, I went on to region C, did the same thing, only dopamine cells were firing, and we had a winner when I measured aggression. The levels of aggression went significantly up, okay? And this is very interesting. This is a region known as the septum, and it's interesting that we found something with this because classic aggression research in cats back in the 1970s, researchers found that damaging this part of the brain would cause the cat to become very aggressive and cause it to hiss and attack. And so it was interesting to find that that was a region that was involved in dopamine's control as well. So from this slide, we can conclude that it's not an overall increase in dopamine that is causing an increase in aggression. It's an increase of dopamine in this circuit acting on the septum that's leading to an increase in aggression, right? That's releasing the mouse's inner ninja. And so on that note, I want to send you guys home with a couple of messages. For one, as I said throughout the talk at different points, the brain is not a bowl of soup, and it's almost a mistake to think that we can treat psychiatric disorders by changing the flavor of the entire soup. Instead, today we have extremely nuanced and powerful techniques that allow us to use just light to control activity in one part of the brain, in just one group of cells, rather than mess the whole thing up. And finally, using this technique and our mutant mice, I'm hoping to further explore how increases in dopamine during adolescence, which is a very sensitive period in development, could potentially permanently rewire our brains to fire more in one particular circuit, leading or predisposing us to be more aggressive in adulthood. And all in all, tying everything together, I can only hope someday, in some small way, my research may contribute to science medicine's understanding of unprovoked and violent aggression, a problem that's so prevalent and relevant to society today. And such that perhaps by identifying one mechanism of what may be happening and where it's happening, we could aid drug development to come up with a solution that reverses the change without producing so many side effects, thus pouring oil into our mental engines just where it's needed. Thank you. Do you guys have questions?
Sure. How do you genetically modify the mice?
So that's a question I never thought I'd get asked. We actually use what we call transgenic mouse lines, and so it's a pretty this is truly challenging my skills to simplify stuff. We use transgenic mouse lines where each cell, different groups of cells, have a promoter or a property that is very specific to a given group of cells, and we use that help me guys if I'm saying anything too. And we use that property to basically drive the expression. So you're basically inserting a gene into this. It's really hard.
So if you think about if there's a gene that's somehow going to encode something specific in a dopamine cell only,
Yeah.
you can take,
And not in any other cell, just dopamine cells.
yeah. So you can take the gene that would encode for just that channel and insert it in there.
Yeah.
So what happens is when a cell is expressing its DNA and becoming a dopamine cell, it's also going to be making this channel.
Okay.
The actual technical way you do it is
Yeah.
beyond.
And so it's basically think of it as just reading all the messages in that DNA, and if we've inserted the one that codes for this channel, you're gonna just
particularly
exactly.
Okay.
And so it just reads all of it together, and just the dopamine cells will have it. And that way we can actually find a property of different types of cells, whether it's serotonin and all these different, and utilize the unique property of that cell, as I said, to drive the expression in just that group. Yeah.
Have you done the manipulation in the adult mice? You have to do it during adolescence?
The drug injections, actually, yes. So what we've done is we've done it at multiple time points, going up to 8 months, and we've actually carved out this tiny window specifically because we did not see changes or increases in aggression when we increased. But that's a really good question. Yeah.
And so what about other neurotransmitters such as serotonin? Is that also involved in aggression?
Yes, there's actually a whole field of aggression researchers who are looking at that as well. And there are different regions that are involved in that as well, but yeah.
So then are these two circuits somehow communicating with each other?
Yeah, so we had some experiments we did because we have a bunch of people in our lab who also work on serotonin, and what we found, interestingly enough, that they kind of work in a bi-directional way, where we increasing dopamine increased aggression, but if we increase serotonin during that same time point, it decreased aggression. The circuit behind that, it's we seem to think that there's some increase in like serotonergic projections to perhaps like the dopamine areas in a way that reduces outgoing activity. So we think that that might have something to do with it, but not looked into it further because that would be 2 PhDs.
This may be a naive neuroscience question, but are there any known differences in the neurons of different regions like the septum that could ever be used to like target drug delivery to just those cells in that region and not the whole brain?
So yeah, it's really hard. I think I don't know enough about like per se how we would translate this in the human. There is a way for us to do it, but that's via surgery in mice because we just deliver it specifically. And like the way we target it is because the hypothesis here is that we're targeting. So when a chemical goes onto a target region, think of it as a lock and key where it just like sets very specifically into a receptor. Okay, and it's like it fits to, and it doesn't fit into all the different types of like locks there are. And so we can develop drugs that are very specifically targeted and inserted just at the region targeting that one type of receptor. And so in mice, that's the way we can achieve specificity. In humans, it's at this point, I think it's a little bit hard to say.
Cool. Thanks again.
Okay, so yeah, now for whoever's interested, we have a tour of our lab. And so, yeah, so we have like different stations, and this is essentially like, if we think about how our experiments go, it's always like, it starts with the behavior in the mouse, and then we get the brain out, and then we cut that brain, and then we take it and look at sections of that brain is the one I showed you of the bright green, and we look at sections under the microscope and make conclusions based on that. And yeah, one member of the lab is going to be around at each station and will answer your questions. So if you guys, I mean, I guess we could all just, I think we're in few enough that we can just all go to one section at a time.
You just can take food or drinks.
Oh yeah.
Yes, or take
Yeah.
pictures.
Or take pictures, yes.
Is there a trash?
Yeah, right there.